In the last video, we explored the transmission theory, what it is, a bit of its history, and some of the medical observations that don't fit neatly into the standard production model of consciousness. Now, whether or not you think the transmission theory holds any weight, what we're about to look at is still worth exploring. Because today we're going to focus on the question, if even part of consciousness is transmitted, what would that signal actually be? And in order to do that, we're going to look at a fascinating paper that not only takes that question seriously, but it brings together a range of experiments that look at how the brain and its individual neurons, how they interact with electromagnetic fields and even light. Now, I learned a lot from reading this publication, but what's more is that it really helped me take a lot of the ideas we've been looking at here on this channel and not only putting them together, but seeing them in a whole new light. So with that in mind, let's get into it. To begin, we're going to do a quick review of the standard model versus the transmissive possibility. I'll put a few chapters in the description below so you can bounce around and get to some other spots if you already know this information. The dominant neuroscience view is that the brain is a productive organ, meaning thoughts, memory, personality, behavior, experience, essentially everything. It's all generated by internal neural activity. The production model is a bottom-up approach where cognition and behavior are reduced to cells, circuits, networks, and neural aggregates. Essentially, the causal locus is fundamentally inside the head. Based on this, the productive model is able to make some predictions that neural activity should correlate with behavior, that stimulating tissue should induce function, that lesions, oppositely, should suppress function, and that ultimately death, which abolishes all activity, means that you are no more. Now, this productive model has been very persuasive. It has over a century of empirical support. Evidence even shows that stimulating the brain and lesion studies give reproducible outcomes. We also know that brain chemistry and electrical manipulation reliably alter cognition and behavior. So it all fits in a bottom-up biological framework. But some examples call this model into question. We review them in more detail in the previous video, which I will link at the end or put in the description. But some of the examples we talked about were hydrocephalus, hemispherectomies, and even psychedelic experiences. There were many examples that pointed to something else might be going on, and the authors, they never claimed throughout the publication that the standard productive model is useless. They are just saying that it may be incomplete, and they offer a new piece to this puzzle. They point out that if even one subset of brain function is non-productive, then a purely productive model is not complete. They offer the transmissive model, which states that something from beyond the brain is received as an external signal. The brain then filters, processes, and modulates that signal, and consciousness emerges through this interaction. Now, no discussion of the transmissive model can be complete without giving credit to William James. He lived from 1842 to 1910. He was a philosopher as well as psychologist, and he contributed greatly to the transmission theory. He has a quote that I really like. "If you wish to upset the law that all crows are black, you mustn't seek to show that no crows are. It is enough to prove one single crow to be white." And this really emphasized what the authors are trying to get across. If even one subset of brain function is non-productive, then the purely productive model is not complete. He likes to use a prism analogy that I think gets the point across pretty well. A prism does not create the colors of the spectrum. Instead, it filters the white light into the constituent wavelengths that already exist independently of the prism. By analogy, he states that the brain might not create consciousness in full, but instead filter or shape it. This model appealed to James partly because it allowed consciousness to be conceived as extending beyond bodily death. Now let's review properties of a transmissive brain, the 4 things it must have in order to be considered a candidate to be a signal here. The first is that it must interact with stimuli whose source is outside the body. Essentially, the source must be extracerebral and it must not be an ordinary sensory input. The signal must directly interact with brain tissue. Second, the receptor structure will have to interact with this signal. Now, it could be a variety of things, such as a conventional molecular complex, like membrane bound receptors, material like structures, such as metals, gases, or interfacial water, antenna like structures involving resonance of cell bodies, or something entirely novel. Third, the signal must physically penetrate the brain and its meninges so that it can interact with the brain tissue. And fourth, it must be converted into neural activity. It could either modulate one region or multiple regions. And after the initial transduction, it may become integrated with ordinary productive neural mechanisms. If it accomplishes these things, it meets the candidacy threshold for transmissive function. The authors argue that existing forces are likely sufficient. You don't need to think of a new force to make this model conceivable. As a result, they focus on electromagnetic fields and optical signals because they can both pass through the skull and interact with tissue. It is pointed out that the transmissive signal may be integrated with other productive mechanisms. Let's take a closer look at this. We already know that the brain is an electromagnetic organ and it generates and responds to electromagnetic phenomena. Neurons are particularly electrically active. The brain contains nearly 100 billion electrically polar neurons. Now, these neurons discharge electromagnetic pulses in the form of action potentials dozens of times a second. Those pulses have detectable electric and magnetic signatures that can be picked up on an EEG. These record what we know as our brainwaves. Interesting story, as I was doing some research, the creator of the EEG, Hans Berger, he was in the military. He had an accident. He thought that the end of his life might be near. And at the same moment, very far away, his sister had a impending sense of doom regarding her brother. She convinced her father that they needed to send a telegram and to check on him to see how he was doing. When he received this telegram, he was shocked that the time when it was created was just about the time of this accident. And after that, he vowed when he was out of the military that he would discover what it was that transmitted from his brain to her brain. And in his search for the answer, he created the EEG. We know a couple interesting things about this, that the average endogenous electric field in the brain is around 2 millivolts per millimeter. Now, that is enough to shift neuronal membrane potential by roughly 0.1 to 1.3 millivolts. Neurons are responsive to sub-millivolt fields down to around 300 microvolts per millimeter. This is significantly below the ambient electric field of the brain itself. This is a big point because it shows that low-frequency fields can modulate brain activity. More than that, the fields created by the brain are not random. The brain's electrical fields are complex, highly ordered. These electromagnetic patterns or fingerprints, these can be associated with cognitive states, senescence, and neuropathology. Because we know electric fields produce magnetic fields, we can also look for magnetic fields within the brain through MEGs or magnetoencephalography. They have found that near pyramidal cells, fields on the order of 5 nanotesla have been detected. And about 1 millimeter from cortical surfaces, bulk magnetic fields are in the 25 to 100 nanotesla range. Outside the skull, they decay very quickly to the pico and femtotesla ranges. These endogenous brain magnetic fields are much weaker than Earth's field. That being said, they still are structured, local, and biologically meaningful. When reading this paper, I came across something new, and it involves Nobel Prize winner Sir Bernard Katz. He lived from 1911 to 2003, and he practiced as a physiologist and biophysicist. He demonstrated that there is a third signaling modality beyond chemical and synaptic signaling. He called it aphatic coupling. Aphatic coupling is a field-mediated interaction between nearby cells. It is described as somewhat analogous to a wireless connection where they interact and communicate with neighboring neurons. It is implicated in olfactory regions, the cerebellum, and hippocampus. In the hippocampus, there are slow, periodic, self-propagating waves that may relate to memory processes. The paper says emphatic coupling likely involves electromagnetic induction, where wire-like geometries of axons and neurites generate electromotive forces in magnetic fields. Even magnetic fields as weak as 5 nanotesla may contribute. It has been hypothesized that glial cells may act as modulators here. Now let's talk about how magnetite may be involved in electromagnetic sensing. Magnetite is an iron oxide mineral that commonly forms crystals. It has been associated with magnetoreception across several organisms such as bacteria, honeybees, pigeons, bats, and possibly humans. Now, magnetite has been found in the brain in shockingly large numbers, in the order of 5 million single domain particles per gram of brain tissue. Deposits of biogenic magnetite have been reported throughout all brain samples tested, and high concentrations have been reported in hippocampal bodies. This is interesting because the hippocampus is involved in memory formation, navigation, environmental coding, and the medial entorhinal cortex contains stellate cells that maintain hippocampal theta rhythms around 7 Hz. We'll see that number come up several more times, 7 Hz. It is suggested that magnetite's ferrimagnetic properties may allow for magnetic hysteresis, which is a material property that can encode and maintain electromagnetic states as a form of memory. Now, the brain is not just electrical and magnetic. It is also optical. The brain can both transduce and emit light directly in the form of ultraviolet, visible, and infrared. These can range from 200 to 800 nanometers and have an intensity of 1 to 1,000 photons per centimeter square. They have been linked overall to oxidative metabolism and neuronal activity. These are also known as ultra-weak photon emissions, or UPEs, and commonly referred to as biophotons. Now, even though the role may still be unclear, part of that is because there's not a ton of research that has been able to be done due to funding. But there still have been experiments that have shown some pretty amazing things. They have shown that when the brain tissue is stimulated with glutamate, which is an excitatory neurotransmitter, that light emission is increased markedly. Evidence now shows that axons may conduct and guide light like a fiber optic cable, potentially increasing the speed and information density of neuronal communication. It has been shown that information can be encoded in direction, amplitude, frequency, and rotational parameters. They think that light may have a similar role inside the brain. We also know that light is capable of entering the skull and that several brain regions express photoreceptors independent of the visual pathway. These non-visual photoreceptors, they transduce or convert specific wavelengths of light into neuronal signaling cascades. You can find these in the cerebral cortices, hypothalamic nuclei, the striatum, the pineal gland, and other regions. 2 examples we'll briefly touch on are opsin 3 as well as opsin 5. Opsin 3, also known as encephalopsin, it absorbs blue-green light below 500 nanometers. When stimulated, it can inhibit neural activity. The image on the upper right is showing the opsin 3 of a mouse. Now, opsin 5 can detect ultraviolet light. The key takeaway here is that the brain emits light, responds to light, and potentially may use light as information. Having looked now at how the brain uses electromagnetic fields and optical signals, let's look at how the brain responds to them in controlled settings. At the microscopic level, neurons have been observed to align and migrate along electric fields in vitro, meaning outside the body, through a process called galvanotaxis. This shows that fields can shape cellular behavior, not just momentary signaling. It has been shown that electromagnetic fields can modulate neuronal activity by affecting calcium flux. Now, this is important because calcium is central to excitability, synaptic transmission, and plasticity. Additionally, electromagnetic fields can also affect microtubule dynamics, which some of you know is proposed to be related to consciousness. In humans, electromagnetic fields have been used to treat disorders of brain function. They can alter perception, modify behavior, and even evoke forced movements paired with a false sense of free will. Now, that last point, I'm going to have to give some thought to. I haven't really gone down the free will rabbit hole. Maybe in the summer, I'll take on that publication related to it, but it's a bit much for now. Either way, we know exposure to intense MRI fields can produce phosphenes, which are flashes of white light, metallic tastes, headaches, and nausea. Many of these symptoms can be explained by activation of the temporal lobes and deeper structures. Now let's touch on TMS, or transcranial magnetic stimulation, which can be used clinically for things like depression, OCD, and other things. Surprisingly, the field strength is 1.5 to 2 Tesla. Now that may not sound like a lot, but for perspective, that's 30 to 40,000 times the strength of Earth's field. Here, rhythmic TMS can cause long-lasting entrainment of brain oscillations. I kind of picture it like an AED for the brain, almost like jumping it and restarting it. It's pretty impressive. I haven't looked much more into it, so if you have any experience with that, let me know in the comments below. We also know that weak fields in the microtesla and nanotesla range still matter because they are used to treat things like brain injury symptoms. They can modulate seizures, inhibit pain, and even induce out-of-body and sensed presence experiences. Additionally, considerable evidence shows that neurons can be activated or deactivated by artificial light. Reported examples include rhythmic light stimulation and training brain oscillations in real time. Transcranial infrared light can modulate alpha power, meaning stronger oscillations in the 8 to 12 hertz range. Light can even facilitate glutamate and dopamine release. Small diameter axons are selectively, rapidly, and reversibly inhibited by infrared light when compared with larger diameter fibers. I propose that this may cause a relaxation effect. The relevance of the transmission model is contingent upon the external signal being present in our environment. And that brings us to Earth's electromagnetic field. Earth's magnetic field averages about 50 microtesla and can vary depending on the time of day as well as latitude. Earth's electric field is about 100 volts per meter and can be disturbed by solar wind and coronal mass ejections. The brain exists within Earth's electromagnetic field, and we know through studies that it can interact with it. For example, there's been studies shown that correlate an increase in geomagnetic activity with increases in psychiatric emission, as well as an increase in seizure frequency for patients who are epileptic. The Earth's electromagnetic field and brain activity can share several properties, including its operating frequencies. Earth's ionosphere cavity supports an intrinsic oscillation at about 7.83 hertz, known as the Schumann resonance, and it has harmonics around 14, 20, 26, and 33 hertz. These frequencies overlap with meaningful brain rhythms, such as theta and alpha. These are important because they affect the hippocampal bodies that allow for memory coding of conscious experience. Several investigations have shown that theta and alpha EEG rhythms can synchronize with geomagnetic fluctuations. In fact, Soroka and colleagues demonstrated real-time coherence between Schumann resonance and EEG frequency spectra across hundreds of brains. In 2019, Kirsch, Fink, and colleagues were wondering whether changes in the EEG could be recorded due to changes in orientation of Earth strength fields. And they found that when people were subjected to the same field strength as, that's around us right now, but they simply changed the orientation, it shifted the state of the alpha rhythms. It has also been found that shielding the brain from electromagnetic fields with copper can selectively attenuate alpha rhythms. Now, sleep may be especially vulnerable. There's an increase noted in vivid, bizarre, even prophetic dreams, and some studies have shown that REM duration can vary with head orientation relative to magnetic north. Lastly, sleep spindles, which resonate between 11 and 15 hertz, they overlap with geomagnetic harmonic ranges. Now we're going to put the pieces together and take a quick look as to what they propose for a possible future experiment in order to test whether the brain has some transmissive functions. And I will tell you before you decide to maybe leave at this point, there's something pretty shocking we're going to talk about in just a moment. We know that the electromagnetic brain interactions that they show a consistent selectivity for the theta alpha frequencies between 4 to 14 hertz. We also know that temporal lobe structures preferentially amplify these frequency ranges. The authors ask whether the selectivity has a material explanation. To test this, they did something pretty wild. They took a post-mortem human brain specimen that was fixed chemically and injected it with current or exposed to time-varying electromagnetic fields. They found that the mesial temporal lobe structures selectively filtered signals with theta amplification between 4 to 7.5 hertz and alpha amplification between 7.5 to 14 hertz. Then they took it farther. They exposed the brains to naturally occurring geomagnetic storm conditions, and they found the right parahippocampal alpha activity was amplified relative to non-storm conditions. This is important because these effects persist after chemical fixation and cell death. So the author suggests electromagnetic brain interactions may not depend entirely on living cell signaling. They hypothesize that brain tissue may passively interact with electromagnetic signals because of intrinsic material properties. Quantitative modeling also predicts that the permeability, which means responsiveness to magnetic fields, and permittivity, which means how it reacts to electric fields, that those of the brain tissue would be around a resonance of 7 hertz. Here, the structural identity of the brain regions may create unique filtering capacities. They specifically speculate that the brain's highly ordered, repetitive microstructure may produce the conditions for metamaterial-like interactions between neural aggregates, especially in temporal lobes. Perhaps the material-based electromagnetic brain transmissive model can excite and interact with select neuronal populations, modulating the productive signal, essentially creating a hybrid effect. It's important to point out that the transmissive model has not been formally tested, but we're going to go over what they propose as to how that may be accomplished. Essentially, brains are a problem for testing because they emit electromagnetic fields and light, meaning their productive functions can obscure the impact of transmission. Essentially, in vivo or in the body, you cannot adequately isolate intracerebral versus extracerebral factors, and tissue material properties cannot be tuned cleanly in living brains. So they have a few in vitro or in the lab solutions that may be able to provide some insights. The paper proposes bioengineering some 3D models, including organoids, spheroids, bioprinted and scaffold-based constructs, as well as brain-on-a-chip specimens. Doing this allows structural tunability, accessibility, tractability, and maximal control. All of this to answer the central question, can information be transmitted to neural tissues using electromagnetic fields and optical signals? Here is a proposed experimental logic. It would be to customize a cell type, something like a dendrite of pyramidal neurons, astrocytes, or microglia, to add a number of cortical layers, to add some extracellular matrix with a composition of a collagen type 4 and proteoglycans perhaps, and then to apply electrical conductivity using synthetic materials such as PEDOT, PSS, and carbon nanowires. Then to expose tissue to electromagnetic frequency sweeps at physiological intensities, all while recording local field potentials. They also mention that Schumann-like patterns could also be applied, Perhaps throw some magnetite in there too and see what happens. Additionally, they propose suppressing endogenous activity with tetrodotoxin and comparing properties before and after chemical fixation. Okay, that is all the fun for today, my friends. Let me know what you think in the comments below. And I really do hope a few of you will read this paper and just let me know what you think. Explore some of the references because there were a ton of good ones. Until next time, stay curious and take care.